our today's speaker, who's Professor Seth Fraden from Brandeis. He has received his degrees from UC Berkeley and Brandeis, where he actually got his PhD, and where he built his beautiful career and an unbelievable research program. He's a true soft matter scientist with an interdisciplinary research at the edges of physics, chemistry, biology, and material science, combining all of that to answer very frontier questions. So he has studied from protein crystallography to making liquid crystalline phases out of viral particles, to DNA origami, to chemical pattern formations, biochemical oscillators, as we will learn today. So again, it's my pleasure to see it.
Thank you very much.
Sorry about
Yes, it's wonderful to be here and see your new facility, and the labs really are awesome. At least I haven't been to the 9th floor, but the 8th floor is awesome. So congratulations on that. I'm sure there's settling in pains, but it's really a wonderful space to have to play in. So today I'm going to take inspiration from nature. So our goal is to make machines that function like this machine, the blue ribbon eel. And so the the thing that you can't take your eye off of is the sinuous motion, mesmerizing. And so what produces that? So I think a kind of way of capturing that all that essence in a static 2D plot is in this space-time plot. So this does not represent 5 different eels, but the same eel at 5 different times. So you think of this as like a multiple exposure from a photograph. And so there's a wave of contraction on the right side, which is kind of represented by this slope in the blue line. So space over time is a velocity, so the slope of this tells you the speed of the contraction down the right side, and then the red line marks out the wave of contraction down the left side. And so in this one static picture that has time in one, it's a multiple exposure, tells you the space-time plot. And so how does the eel generate this space-time pattern, the spatial temporal dynamics? So the neuroscientists take the eel while it's alive, they flail its skin off its bones, stick electrodes down its spine. Here's 8 electrodes on the left side, 8 electrodes on the right side, and then like this line here is this is a double space-time plot. So the blue is the electrical recordings from this electrode, the red are the electrical recordings from this electrode. And so as you go from head to tail, you see this wave of activity, of electrical activity down the right side, and then a wave of electrical activity down the left side in this space-time plot. So the view is that the eel generates, the neural tissue generates the spatial temporal pattern, which then of electrical activity, which innervates the musculature and causes, this marvelous motion. So then neuroscientists say, make a minimum model of neural architecture that could account for this. So each of these circles represents a self-driven oscillator. So you say the neuron is something, an individual neuron produces periodic spikes of electrical activity, and then the arrows with the head on it mean they're activator coupled, meaning if this guy fires, it wants to make that guy fire. Bidirectional means that, likewise, when this guy fires, it wants to make that one fire. And then the hammerhead things look like a nail with a head and a tail, that means inhibitory coupled. So when this guy fires, it wants to inhibit that one firing. So the neuroscientists, when they cut open and dissect the eel, they see this kind of neural network, where they're oscillating neurons, laterally inhibitory coupled, and then longitudinally excitatory coupled. So we want to make we want to make robots like this, using these kinds of architectural design principles.
If all the couplings are bidirectional, why is there a direction the way it propagates?
That's a good question. And one could say spontaneous symmetry breaking, but you could also imagine that you've got like a one head neuron that is the master and falls but that's a good question. So the kind of conceptual framework to understand this we're going to adopt is the discrete reaction diffusion model. So where this C is the vector of M chemicals, to make an oscillating reaction, you need some set of chemicals. These F's are the rate equations, nonlinear rate equations that govern the behavior of a single oscillatory. And then this is the coupling, that tells you is reactor I connected to J or to something else that's encoded. This is it's called the network of Laplacian. It tells whether you're connected or not to a neighbor. The D is a diffusion matrix, which tells how much of one species communicates to another. And then we're assuming that it's Fick's law, that the flux of chemicals between connected wells is proportional to the concentration difference of that species. So just Fick's first law, J equals minus D grad C. So, this is the physics, this is the chemistry. And so this is the reaction term, this is the diffusion term. So reaction diffusion network dynamics, we're assuming all the chemistry takes place at a point. No chemistry takes place on the connection in between. The connection in between is only the transfer of molecules. The reactions occur without any spatial dimension, just as a point. So this is the simplified model first introduced by Turing in his 1952 paper, The Chemical Basis of Morphogenesis. Okay, so in order to get a neural tissue, the properties of a neural tissue, the organized spatial temporal patterns, we need to have either oscillatory or excitatory dynamics. The F has to be, the nonlinear chemical equations have to be complex enough or rich enough to have either oscillatory or excitatory dynamics. We need a complex enough network topology. If you just have two oscillators, that's not enough. And then we need to have a coupling matrix that can either be excitatory or inhibitory. And in some cases, in order to answer your question about how do we make it directed, we also need to build in directed networks. So these are, and that would be encoded into this matrix here. So, so these are the kinds of things that we're going to need in order to experimentally realize this plan. So our model system, the fruit fly of nonlinear chemical dynamics, it's not a fruit fly. It's the Belousov-Jabotinsky oscillating chemical reaction. When it's clear, that's in its oxidized state, and when it's red, it's in this reduced state. So this is a ruthenium 2+ when it's like that, and a ruthenium 3+ when it's like that. So this stirred beaker means there's no concentration gradients. All the concentrations are the same everywhere in space, so this is a point chemical reactor. So this is one of our points in this diagram. Yes?
Is this real time or is it sped up?
So this is sped up, but I can make the reaction 10 seconds if I want. I mean, I do hands-on experiments for high school, and we can make it 10 seconds. So depending on the concentration, the pH and the concentration of sulfuric acid, we can tune it from a second to an hour. So but this is a closed system. It will oscillate 100 times before it dies. So if I do it a second, then it's over really quickly. And so we tend to make them 10 seconds to a minute. Okay, so then also taking a dynamical systems perspective is very useful. And so you see there's this limit cycle behavior. So here I'm plotting the inhibitor, activator, and catalyst in this plot. And then now I just take each of these axes, each of these species, the catalyst, the inhibitor, and the activator, and I plot them as a function of time. So the limit cycle means that no matter kind of where I start in this this space of possibilities of chemical concentrations, it evolves to this limit cycle. And you can take a very reductionist view where you just focus on two elements. One is the activator. So at a certain amount of time, the reaction turns on and then slowly turns off. And when the activator turns on, it activates the production of all the chemicals, including the inhibitor. And so then as the inhibitor builds up, it eventually gets to a concentration at a later time that starts applying negative feedback to the activator and turns it off. And then there's a third reaction that eats the inhibitor. And the inhibitor gets consumed over time, and once the inhibitor falls to a low enough level, the activator can go again. And so you get the cyclic process of activation and inhibition. And for the purposes of this talk, we're going to use materials that exploit these physical properties. The inhibitor is a neutral molecule, bromine, and has a low dielectric constant, while the activator, bromous acid, is charged and has a high dielectric constant. And we're going to exploit these physical properties of the inhibitor and activator in order to make semi-permeable membranes that let one species go but not the other. So then, nonlinear dynamics 101 in one slide. To get an oscillator, you need to have a certain complexity in your activator and inhibitor. And so if F is the nonlinear equation of the two species, activator X, inhibitor Y, F tells you it's how the change in the concentration of the activator varies in time. And this line here is the Knoll line. So above it, the activator is decreasing in concentration, X dot is negative, and below it it's positive. And likewise, this is the Knoll line for the inhibitor. On this side, the inhibitor is decaying. On this side, the inhibitor is growing. So how does this lead to an oscillation? If the steady state where the two activator is zero, the change in activator concentration and the change of activator and inhibitor are zero is where they cross, then if that's on this rising part of the cubic, then you can get an oscillator. So what happens? Say I'm in this region. In this region, both X dot and Y dot, the inhibitor and activator, are increasing. So I get this sloped line. When I hit this line, then this is the activator has zero time dependence. Then but my inhibitor grows, I start moving up. But as I move up, my activator in this region decreases. So then I move back to the line. And so I just hug this line until I get to this point. And when I get to this point, I move off the line. And once I cross over here, both are negative, both rates are negative, and I zoom over to this line, and then I hang on this line. And so if I have a cubic equation like this and an intersection like this, I'm going to get an oscillator. So this is how you get a limit cycle. And kind of the breadth of the limit cycle then depends on how wide apart these branches are, and and this is the chemistry. So all the chemistry goes into designing these rate equations so that you get one is kind of linear and one is cubic, and that you adjust the intersection, then you make a chemical clock. So that's what chemists do. So these are the principles they use. My colleague Irv Epstein at Brandeis has been working 40 years on this, on building chemical clocks. If you don't stir, then you get these chemical waves. And so the blue is the oxidized, where and then the red is the reduced. And you can see how nonlinear it is, because when two waves intersect, they annihilate. And that's because just after you've activated it, there's a lot of inhibitor produced. So on the backside of the inhibitor on the backside of the activation front is a large inhibition. So then it annihilates the oncoming wave. Okay, so that's all the chemistry that we're going to focus on. So what about, we're not going to make a fish out of Erlenmeyer flasks. You know, that's too big an object. So we have to reduce them. So we use microfluidics to make these little drops. And then we put them up into arrays, and then they oscillate. And so here we're shining light from behind. And when it's in oxidized state, light is transmitted through. And when it's in the reduced state, it's not. And so, you know, we build these kinds of networks. We can add a light-sensitive catalyst. So now we're shining blue light onto these with enough intensity to suppress them. This one gets a little bit of intensity. You can kind of see the dot is smaller. And so it oscillates, but slower than the one that has no light on it. And so I kind of use light like you do in optogenetics to turn on and turn off the neurons, you know, if you want to make the fiction that these are, you know, like neurons, these nonlinear oscillators. And so you have a computer projector and a microscope, and they're different wavelengths of light, and so you can filter out the computer, you know, and so then you don't see, you know, this is actually being illuminated. All the drops except for that one are being illuminated, but we can filter that out because one's blue and one's green, so that you only see the state of the chemical system and not your perturbation. And so you can control these things. This is the scary slide for the physicist. You know, so there's like, you know, 10 rate constants. Yeah, there's number 10. This one's not numbered, so that's number 11. You know, there's a ton of rate constants. We don't touch any of them. We don't fudge any of them. It's because we wouldn't even know how to fudge them. You look at this, it's kind of, you know, just too complex to wrap your head around. And so, but the chemists have studied this for 4 years. They've measured those things, and they just say, and we just use them. And they say, okay, you can vary the concentration between 100 millimolar and a molar, and they're good. If you use 10 millimolar, you know, we take no responsibility. If you use 2 molar, you know, you're on your own. But they say within this set of parameters, you know, we stand by these constants. And so they're not fudge factors. They're just numbers. We put them in MATLAB. It's like it's the engine under the hood. You know, we don't look under the hood. I mean, I have no idea what any of these things mean, really. And it's, to me, it's unimaginably complex. And, you know, but these are not fitting parameters. These are just numbers. Okay, so then how do you make something simple out of this? So what the dynamical systems people do is they say you have a limit cycle. The limit cycle is a one-dimensional manifold in this m-dimensional chemical space. So you can reduce the problem to this one-dimensional problem by saying that you only have one parameter, the phase of the chemical cycle. And then that tells you uniquely what your chemical composition is. And so if you have a sinusoidal activator as a function of time, then then you would have these uniformly spaced isochrons, lines of equal phase, just like the hands on a clock. And what an isochron is say I start at this time, where this line here, then 1, 2, 3.5 hours later, I'm here. And if I start at this point means I have higher inhibitor and lower activator. At 3.5 hours later, I'm at the same point, because every point in this phase plot has to end up on the limit cycle. And therefore, there has to be a mapping from the m-dimensional, you know, space of the chemicals to the one-dimensional space of the limit cycle. And so there's so any point along this isochron, these are lines of equal phase, because no matter where, if I start anywhere on this line, I end up at the same point on the limit cycle. And so this concept of isochrons is a fundamental concept in nonlinear dynamics. And so it's saying the limit cycle is like a clock. If the variation is sinusoidal, then it's a regularly spaced clock. Now, this is kind of what the limit cycle looks like for the BZ reaction. So the lines are very squinched in here. That means that you get this very sharp, relaxation oscillator type behavior, where you get this very short oxidation spike, and then it spends a long time with low activator concentration, and boom, it activates very rapidly and then comes back and spends a long time in this reduced state. And so it's still a clock, but it's just time is very warped, but it's still a limit cycle and a clock. Okay, so then what happens when you perturb it? So here's my inhibitor, here's my activator, and now I add a little perturbation. So I add a little bit of inhibitor. So you can see I pushed it forward. It was in between these two isochrons. I gave it a little bit of inhibitor. Now it's up here on this isochron. And so if I let it relax to the limit cycle, and I compare it with where it would be if I had not perturbed it, it would have been here. Now I instantaneously advanced it by half a isochron, so it's half an isochron ahead of this. So this is a phase advance. I gave it a little perturbation, a little bit of light that increased the inhibitor, and if I had given nothing, it would have oscillated at these dashed lines, but it advanced a little bit because I gave it light, I exposed it to light at this moment. If I gave it the same amount of light up on this part, now it moves back half an isochron. And so it would be a delay. So compared to if I did not perturb it, it is behind it. So the same dose, the same amount of inhibitor has a very different response depending where you are. And we all know this from jet lag. If we fly to Europe and then you get up in the morning and you go out in the sun, then you can get phase shift forward. But if you do the same thing coming back this way, then you'll have jet lag. You'll advance, and then take you 3 days to come back to your steady state. So the same exposure can have different outcomes depending where you are on your limit cycle. Okay, and then just for nomenclature, I'm going to use this kind of vector notation where X is all the chemicals, and then f of X is a vector equation of all the chemicals, and then G is my time-dependent perturbation. So then experimentally, we here's 16 experiments being done simultaneously. The illuminated drops are knocked out, and so they isolate the ones that aren't illuminated. Then we wait for the drop to spike twice so that we know its period, and then we give it a perturbation at some known time and then we look if it was not perturbed, it would have spiked at this, and then we measure the time delay induced by the perturbation, and we do that as a function of the phase of the drop. Okay, so this is the phase response curve, and so your response is different depending on when in your cycle you get perturbed. So if you're just about to spike and you add a little bit of inhibitor, it delays you a lot. While if you have spiked and you're pumped, your reaction is super high in inhibitor, adding the same amount of inhibitor does nothing. And so there's a refractory period. In the first half of the period, you're insensitive to the perturbation. In the second half of the period, you're very, very sensitive to the perturbation. And and this light pulse is only inhibitory, because the light couples to the inhibitory mechanism. So we can measure this phase response curve. Okay, so then what's the math of a weakly perturbed oscillator? So this is due to Kuramoto in the 70s. So, so we want to take this chemical, here it's only 2 dimensions. In our model, it's much higher number of dimensions. We want to take the m dimensions of the chemical limit cycle and reduce it to 1. So this is the change in phase with time. This is the frequency that you make 1 revolution of this limit cycle. And so we use the chain rule to break it into the phase response curve, the change in phase with added a little bit of perturbation. That's this thing we measured in the previous slide with the light applied to the individual oscillators. And then dx/dt is the dynamics of x is the chemical concentrations. That's the dynamics given by this equation. So we can just substitute in the phase response curve for an individual oscillator with the dynamics of the chemical from this equation. And then Q times F is the frequency of a single unperturbed oscillator. And then this tells you how much the phase is shifted by the perturbation. So it depends only on the single particle response function and the amount of perturbation. So in this model, this Kuramoto model of weakly coupled oscillators, you can tease apart the you can express the response in terms of you can decompose the perturbation into a susceptibility function and the perturbation. So this is a very powerful method. And so this G, in the previous slide it was light that we were applying, but this flux can have any origin. It can also be the flux from a neighbor. So every time a neighbor oscillates, it produces inhibitor which will diffuse across. And if we have 10 neighbors around it, then since this is linear, we can just sum up over the 10. And so if we want to build the interactions of one oscillator in the presence of any number of other oscillators, we can use this formalism because this perturbation could be anything, including the contributions from many neighbors. So now let's take a look at the contribution of this neighbor on this neighbor. So here we have a capillary, like 100 micron diameter glass capillary. We have a drop of the BZ here, a drop of the BZ here, oil, fluorinated oil in between. And so every time this one oscillates, it sends out an inhibitor to this one, and vice versa. And we can model this with finite element, where here we've colored the inhibitor, and this is the BZ drop, and then this is the flux of the inhibitor, and we know the partition coefficients, and so we can calculate this exchange, and then we see the drops in the model are going 180 degrees out of phase with each other, just like the drops in experiment. And so you have these self-driven oscillators. Each drop wants to oscillate, but each drop also wants to prevent a neighbor from oscillating. So you have inhibitory coupling, and it's symmetric. And that leads to going 180 degrees out of phase. And then this is the rate, the diffusive flux, the diffusive rate of molecules going from one side to the other. And it depends on the area in between them. It depends on the gap in between them, and it depends on the volume of the receptacle. So what's going on here? So imagine I have 2 drops, and this drop is at this phase, and this drop is at this phase. Now this drop is higher in inhibitor than this drop. So there's going to be a concentration flux of inhibitor from this drop to that drop, just like shown in this picture. So this one's going to decrease its inhibitor concentration. This one's going to increase its inhibitor concentration according to the phase response curve of how much the phase shifts with a unit dose of the chemical species. And so you can calculate analytically this response. This is this so-called Kuramoto phase model, where you have a phase-dependent interaction function. And then that tells you how much this drop changes in phase with respect to that one. And so Kuramoto, you say, okay, I have my phase response from a single drop, and now, since I have drops at different, that are at different phases in their oscillations, this one is at a different phase than that one, they have different concentrations, there's going to be a flux. Fick's first law, the flux is proportional to the difference in the concentrations times the rate of transport. That's just this geometric factor that depends on the gap and the area. And then if I integrate over one period, this tells me how much I shift phase averaged over one period. So now I have something that's time, that doesn't depend on where I am in the period. So you have the fast dynamics of a single oscillation and then the slow dynamics of the whole system evolving to its steady state. So this is a separation into two different time scales to simplify the problem. And so then where this is the phase response curve, and so then this is the drop-drop coupling. So this is how much one drop influences a neighboring drop over per cycle, depending on how different those drops are in phase. So this is the, and so this you couple, this you calculate completely from the higher level theory. So where the chemists have spent 40 years validating all the rate constants. You take all that information, but that was a m-dimensional problem that, you know, is too ugly to even think about. And then you can beat it down to this one-dimensional problem. Well, here's two-dimensional because I have two drops. But then we can go further and we can beat it down. So this is where we were with the, where we have one equation for each drop. But then if we look just at the phase difference, now instead of having this 2m dimensional space, we just have a one-dimensional problem on the phase difference between the two drops. So I want to know, do the two drops synchronize? I don't care if they synchronize at 3 o'clock or at 12 o'clock. I just want to know what their relative phase difference is. And so then I just have this one equation. This is this coupling equation of the differences. And then I can just use one-dimensional stability analysis. So if my phase difference, this tells me how the phase difference evolves in time, the time derivative of the phase difference. The interaction function, the effective interaction function is positive, so that means that that this increases until it hits this point. Over here, if my phase is bigger, then it decreases, so there's a fixed point here at pi. And so you've proven that if I have two drops with any kind of inhibitory coupling, they're going to synchronize at pi. So you've gone from this, 2m dimensional space to a one-dimensional space where you can really visualize it. You can separate what is the chemistry, what is the physics, what is the role of topology, what is the role of chemistry, and understand the dynamics. So this kind of framework of introducing this phase model when you have a limit cycle is a very, very powerful method, and the neuroscientists exploit this, very heavily to make progress in understanding, neural networks. Okay, so that was two drops. Let's do dendrites. So this is the star. We're going to have a central hub and then arms. So we make this out of a silicon chip so that if we put a drop here, it can't communicate with that drop because there's impermeable silicon here. And then we take the drops and we load them into these silicon chips. So you just take a drop of this emulsion, you plunk it onto this chip. This chip has thousands of these wells, and then you flop down a cover glass. It takes like all of 10 seconds. And with something that crude, you get a tremendous amount of failures. So like here you see there's no drops there. There's 3 drops here. This one has 4 drops, and this one failed. But there's some that are successful. And so if you get like 10, 20% successful, then, so out of several hundreds, you get 20 or 30, but it only costs you like 10 seconds to make the sample. The graduate students prefer that than to spend hours making a sample, that you get a few more. And so, and then these are gutters in between each one where the excess drops can fall off, and then when the glass slide comes down on top and seals them, each one of these is independent, and then these drops are down in the gutter. So they're oscillating, but their chemicals can't affect these chemicals because they're in sealed little wells. Okay, and so then what's the phenomenology? So if you have long arms, then they just flash at random, and you see unlocked. This is the number of arms, star degree. This is how far apart they are. So you get an unlocked region when they're very far apart, and it's unlocked. And then this regime, which is the most interesting, is
This, what are you pointing to?
this regime here is the most interesting. So these 4 arms are locked, and they're out of phase with the one in the center. So this goes beep, bop, beep, bop, beep, bop. And it's not 180 degrees out of phase. When this guy oscillates, these guys oscillate rapidly after. Then there's a long pause, and then this guy oscillates. So they lock, but not at 180 degrees. And now if I add one more arm, this has 4 arms, this has 5, but they're equally close, then what happens is there's so much inhibition coming into the center one that this one dies. And then when this one dies, these ones which were locked become unlocked. It's just random. So when this one dies, there's still chemical reactions going on. It's just not on the limit cycle, but it's at a locked state. So if I add a little bit of inhibitor, the chemistry gobbles it up because it's at a stationary state. So it's like a voltage clamp. You know, if I have a ground and I insert charge, the ground bleeds those charges off to earth, and the voltage stays the same. Same in here. The chemical concentrations in the center stay constant. Even if I feed in inhibitor from the arms, it gobbles it up and maintains it at a constant chemical. And so then these arms don't communicate, and they're randomly oriented. So I want to focus on why is this one not at 180 degrees? What governs the phase shift between the arms and the center? If I just have 2 of them, so then this is a star arm of 3, and I see this would be pi, and the phase difference between the 3 arms and the center, there's some transient behavior, but it locks at some angle that's not pi. And so if so you have your single oscillator, and then you have the sum of inhibition coming in from all the arms. And so, so these are the rate equations. 4 of them for each of the arms, so there's 12 of these equations that then, that feed into this one. And then we have inhibitory coupling, so only the inhibitory species is coming across of the 4 in this term here. So each reactor has all the chemicals, but the crosstalk is only via inhibition. And so if you just have the arm with 2, beep, bop, beep, bop, beep, bop, you get a 180 degree phase shift. And then as you add more arms, the phase shifts systematically deviates. And, and then when you have 4 arms, it's this asymmetric pattern. So, so it's like 1 beats, then 4 beats, 1 beats, then 4 beats, 1 beats, then 4 beats closely, while when it's 2, it's equally spaced. So if you look at the kind of full dynamics in the phase difference space, no matter where you start, you find, this is a numerical model, you go to 1 single attractor. So that suggests that it's like 2 oscillators. You kind of think all 4 arms are synchronized. It could be, you could conceptually imagine all 4 to be just a single oscillator that's 4 times bigger. Because each one gives a little dollop of inhibitor. There's 4 of them, they're adding up. So just think of it as a 4 times bigger oscillator. So you think of it as a small one in the center with 1 on the outside that's 3 times bigger. And so then you reduce your problem to one of 2 oscillators. And so then when you go through and you do your coupling now, when it was, this is k is the number of arms. So when there was only one arm, so it was two of them, then you had this symmetric function, it would intersect at pi. But as you add more and more arms, this function becomes asymmetric, and the intersection, the stable point, shifts. And so it shifts off of pi. And so this is a numeric result, and then you plot it kind of on this log scale, and this is the experiment, and then this is this phase model. And it suggests that that the phase shift goes like log k. And so then you can kind of then make a guess that you'll do an expansion of this locking angle in a series where the small parameter is log k. Right? So this was actually suggested to us by Michael Rubenstein. He visited the lab like a month ago and said, and I thought, oh, that's just the craziest thing. Who expands in log k? That's stupid. And then the postdoc does it, and it just worked perfectly. And then the postdoc, and you get this analytic expression. And then the postdoc says it had to be log k because, let me go back. Okay, so you have this small guy and this big guy, and you get a certain phase shift. You don't know what it is. Say it's 20 degrees. But now let's say I interchanged them, and I made this guy the big guy and this small one. Then it would have to be the same magnitude of phase shift. It's the same system. I've just rotated it. But then it would be 20 degrees in the other way. And so instead of pi, it would be pi minus delta. And if it's 1 to 4, it's pi minus delta. If it's 4 over 1, it's right. So if it's 4 over 1, then the answer is pi minus delta. If it's 1 over 4, then it's pi plus delta. Well, what function gives you a minus increment when you invert it? It's only the log. The log's the only function. Log of 1 over x is minus log of x. And so the postdoc goes, it had to be log. Anyway, so I guess Rubenstein, he didn't. So I thought, who does Taylor expansion in log k? That's ridiculous. Anyway, so then you get an analytic expression, and you go through it all, and then you get this analytic expression where this part is the topology, the log k and the pi comes from topology, and then all the chemistry is here. And so this approach really lets you separate what aspects of the dynamical systems is from chemistry and what parts are from topology. So I thought this was just kind of, and everything is completely controlled. This H function is calculated from this phase response curve, which in turn is calculated from the full chemical dynamics. So there's like 0 adjustable parameters in this. And so what kind of questions? So if you have this kind of experimental system, what kinds of things can you do? So we view this as like a model nonlinear chemical dynamical system. What kinds of experiments, what kind of questions can you do, can you address? So one is the fish one, the eel one. Given a network, what kind of spatial temporal patterns arise? How many attractors? What are their basin of attraction? And then what happens when you vary topology, interaction strength? If you make the connections inhibitory or excitatory, directed versus undirected? If you allow phase shifts between the connections or heterogeneity? So we can explore these kind of fundamental questions. Other questions are, can we tease apart what aspects arise from the chemistry and what from the topology? Because then we can build intuition and understand design principles for creating networks that have functionality. Sometimes with photolithography, it's very easy to vary the topology. Or by temperature or light, you can change the chemistry globally very easily. Or optochemistry, you can change some chemicals or not others. So so being able to tease apart the contributions from topology and chemistry really expands your design set. And then, can we make useful materials? Materials that are capable of locomotion, like swimming and running, or mass transport, like the pumping of your heart. Can we make soft robots? And so those are the kinds of questions we're using. So we have different projects that are focused on each of those questions. And so to make the soft robot, we have to make materials that are inhibitorily coupled. So that's this array. Every one of these is inhibitorily coupled. And so in the space-time plot, you start them all in phase as an initial condition, but then they evolve to be out of phase. So like this red line here is, we're digitizing the light intensity along this red line and then plotting it as a function of time in the space-time plot. So the spatial history of this one well is here. The spatial history of this well is here. So you see, after a while, they get 180 degrees out of phase. When this guy oscillates, the two neighbors are 180 degrees out of phase. When this guy oscillates, its neighbors are out of phase. And so, but here we've made little channels between all of the wells. And so then when you follow this one as a function of time, then you see that they stay in phase, and so they're excitatory coupled. And so there's these little tiny channels that let the activator come across. Here there's no such channel, only the inhibitor comes across. And so with photolithography, we can make arbitrary networks where we can choose to make them excitatory coupled or inhibitory coupled. And then we can measure, so the neuroscientists say this is the circuit, which we reduce even further. They say inhibitory coupled across the spine, excitatory coupled along, we reduce it even further. And so we make, this is one side of the spine, and then we shine light on all of these so that they only are excitable. They're not oscillatory. And then this one is darker, it's oscillatory. And so this is the pacemaker. And then to control against any experimental gradients in the system, we just shine light so that all these are excitatory and only this one is oscillatory. And then we measure the wave speed in each of these and take the average. And then this is one without any nodes to just kind of be a control. And then we see in the space-time plot this kind of fast, slow, fast, slow, fast, slow. It goes slow through the little narrow and fast through the wide thing. And then we can model this in finite element, and we see it goes really fast through the wide and really slow through the narrow. And we can explain this from kind of first principles of chemical waves. And we can compare theory and experiment. So we kind of understand the excitatory coupling, we understand the inhibitory coupling, so then we can put them together. So here we have our ground plane, so this is the constant chemical being shown with invisible light. We put our pacemaker here in the dark. We set them off in phase as far away from the design attractor as possible. They're inhibitory coupled this way, excitatory coupled that way. And then this is the data from this experiment. This is the space-time plot. So this is the time dependence of these two. This is the time dependence of these two. And then if you look at the phase difference as a function of time, bam, right to the attractor. Depending on the gap, you'll get more or less robust behavior, statistics on the robustness of the circuit. So now you want to make your robot. So then you have your shape-changing polymer where the reaction is confined to the polymer because you put the catalyst on the backbone of the polymer, and then in the next step, we haven't done this yet, then we're going to laminate them together to make the fish. So this is what we've done in the last year, is we've worked out that we can control network topology, we can control initial conditions with light, boundary conditions with either using the constant chemical boundary condition with the light on the chemical reaction, or no flux boundary condition using the silicon. We can vary the reactor volume, the coupling strength, and we can make whether or not it's excitatory or inhibitory. And so our two kind of principal aims are to tweeze apart the topological and chemical contributions and to fabricate synthetic materials that produce spatial temporal patterns with the functionality of living neural tissue. Thank you. And most of the work was done by this talented postdoc who's on the faculty job market, and like Norton, who is a theorist.
Questions?
So, of all the oscillating chemical reactions, you chose BZ.
Yeah.
Is there a reason?
The 100. So it will oscillate 100 times before dying. So it's the longest of all known chemical reactions. So I don't have to make open reactors to feed in. So I tried making open reactors with the microfluidics. It was a nightmare. And so the BZ, also the BZ, they've been studying it since the 60s. They know, I'm not going to say they know everything about it, but they know a lot.
So what goes wrong with it? Does something oxidize, or?
I told this to the grad students. It's no different than the grad student. Like if you lose your grants and you can't pay the salary of your grad student, they'll come the next day with no pay. They'll be hungry and they'll work. They'll come the next day. After like 5 days, they'll start, getting, kind of comatose. After a week, they'll pass out. After 3 weeks, they'll die.
They'll find free food forever.
Huh?
They'll find free food forever.
If they're starving, they're gonna die. If you don't feed them, they'll die. They'll come a few times out of habit, 10, 20 times. BZ's no different. So we loaded with fuel, malonic acid. It's basically gasoline, and bromous acid. And every time it does a cycle, it cracks the malonic acid into bromomalonic and produces CO2, just like the graduate students. They breathe in oxygen, out CO2, they eat their pizza, they excrete pizza, or some waste product. And, so it's so you're consuming mass and energy, and it's a closed system. You can only do it so many times. 100 is the best that we can do. Your graduate students, I don't think they'll come in 100 days working.
Were those the bubbles that you could see?
So we pressurize the system. Otherwise, we produce the CO2. If we don't clamp the system rigidly and seal it, then after several oscillations, then you'll start building up CO2 gas, and then those will plug the channels and things up. So you have to carefully seal so that it pressurizes. So it's like a Coke bottle, like a beer, bottle. When you open it, then the CO2 nucleates and it fizzes, like it's dissolved in no beer under pressure. So we're doing the same thing here. We're pressurizing to prevent the bubbles.
This is really cool. This is probably a stupid question, but why would you use this to actuate soft robots when electronics go more than 100 oscillations?
So I wrote a paper, the first paragraph is like, this system is 10 to 11 times slower than a computer. It does, it's 10 orders of magnitude worse in any possible way. No question about it. But these are the reaction to fusion is how you work, and you build computers, and you operate them, and you can walk around and scavenge pizza, from free food, and whatever. And so the fact that we're here talking to each other means that with reaction to fusion, you can do kind of useful things that you can't do with the robots we make now. And so life appeared on the planet around year 500 million, plus or minus 200 million. They don't know. So let's just call it 500 million. The first nervous system arose 3.5 billion years later. Okay, but then when it did arise, virtually every multicellular organism uses them. And so clearly, two things you learn from this. It's hard to make spatial temporal patterns that organize chemical activity, and two, if you can make it, it's really useful. So NSS has been funding us for like 10 years to do this. Imagine they just funded us for 1,000th of the time it took nature, just 1,000,000 years, you know, you know, even 10 years I would settle for, you know, 10 to the,
But why,
you know,
why
10 to the
is-
7 times less than.
But should we assume these have intrinsic advantages, like steel is better than bone, right? Like there are
Is
things
it?
that nature can't
Is it?
provide.
How
Well,
many
no,
how
in
many
many
steel
ways.
things repair themselves?
Right.
But
But
you
like for
break a
certain
steel
purposes-
beam. For certain purposes, steel is better than bone. For certain purposes, silicon computers are better than parallel computing, you know, of the brain. Yeah. So there are, but then there's other purposes that, you know. And so it's that kind of attitude. Why would one pursue this? It's like, you know, that blue ribbon eel is pretty cool. It's got this really cool dynamics, and none of our artificial, you know, and so it's just, you know, can we, you know, so I know that you can do cool things with reaction diffusion. You know, I know it took nature 3.5 billion years. I think we can do it, you know, in order of some tens of years. You know, we, the engineering, you know, soft matter science community. And then I think one could then take the best of both worlds, the robotic, mechanical, CPU world, and then the soft matter world, and then cyborg, what's the Star Trek, what's the appropriate sci-fi, you know, hybrid brain electronic neural interface, you know, dysfunctional universe that I'm imagining. But yeah.
Andrew.
So does this, how far does this scale? Because the things that you were showing were, I mean, had a number of
Yeah.
elements.
Yes. So, you know, how big are your cells? You know, they don't get bigger than 100 microns. I mean, like the world's biggest cell is the frog oocyte, 1 millimeter or something. But, you know, typically cells are 10 to 100 microns. And so that's how far it scales. I mean, these things are no different, because if it's diffusion, then the time goes like L squared, you know, and so, you know, you're not going to go from 100 microns to a millimeter. That's 100 times slower. Forget it, game over. So just like we're made from little cells that then are integrated into tissue, and then tissue into organs, and then organs into organism, you too would, your engineering principles would be also this hierarchical scale, where you would have, you know, microfluidic channels innervating the flow in, you know, with these string together elements, you know, and so your gel would be a bunch of little gel beads, you know, and then each gel bead would contract, but then, you know, you need strings of them in order to, you know, just like your muscle fibers. It's that the reason we're built this way is the same kind of flux chemical engineering. Those scaling laws that apply to biology will apply here as well, because it's the same physics, it's reaction diffusion.
Mark, you have a question?
Yeah. Do you have to break detailed balance in order to get directional coupling, or is there something?
No, you don't. And so we have some schemes for the directional. So it's kind of related to that chemical wave thing. So when you go from a small orifice to a wide one, then the chemical wave curves, and then as you have a concentration front that's expanding, it dilutes. And so that can be a negative feedback. And while you won't get that from a large to a small, and so you can break symmetry that way by using the expanding wavefront. You make chemical diodes this way. So we're working on that now. Yeah. So the chemical diode. Yeah. So you don't need to break detailed balance for a chemical diode.
Adrian?
So basically, this is a propagation of an information. Right?
Yes. Yes.
You have been saying about the activation. I mean, how do you translate this information into an activation?
Yeah, so that's a really profound question, and it's one we're actually also trying to understand. Is, you know, can one put this in an information framework, and what would be the information carry, so for Shannon entropy, it's very clear what a bit is of information, but for chemical reactions, this is an area of theoretical activity, especially like in synthetic biology, and trying to try to quantify those notions of information-rich reactions or not. And so this is something we're following the literature in this area, but I suspect that maybe we can test some of the new theories that are coming out with this system, but this is very much at the forefront. I mean, that's a really forefront question, is what's information content in a chemical reaction? And that's something we're very interested in. So if you have any suggestions, I'm open to them.
Any more questions? If not, let's thank Seth again.